In this video, we're going to look at DNA mutations. Because there's so many possibilities for cells to become mutated, it's important to see how those mutations can have an impact on our cellular function. So remember that DNA codes for an RNA molecule that codes for a protein. If the DNA has a mutation, what will happen to the RNA and the protein? They could have mutations as well. If a protein doesn't do its normal function, let's suppose it's a membrane protein that transports chloride ions. If that doesn't function and do its job, then the cell can't move chloride ions, and then you have cystic fibrosis. So in this video, we'll look at the different types of mutations, where do they come from, and then what does our body do about them? Most DNA mutations are completely random. There's no specific gene in any specific cell that's targeted. When substances in the environment impact our DNA, or when DNA replication mistakes occur, it is usually completely random. But let's think about the different cell types we have. We have germline cells that are going to become sperm and eggs. That DNA gets passed on to the next generation. And then we have our somatic cells, which is our body cells, like our skin and our liver and our blood cells. If we have a mutation in a somatic cell, it is only going to affect our body in this lifespan. If we have mutations in germline cells, they can be passed on to future generations. So DNA mutations can have a very huge impact on evolution. In fact, DNA mutations are exactly where every living thing came from changes in the DNA sequence. Any DNA mutation that's not detrimental can survive in a population, and it will increase genetic variation. In populations, having genetic variation is very important for survival. So let's look at the types of DNA mutations. There are two major categories of mutations. We can look at point mutations and recombination mutations. Let's suppose we have this normal DNA gene sequence up here. DNA is double-stranded. Let's suppose this bottom strand is going to be the template for RNA polymerase that will produce an RNA molecule, a messenger RNA molecule. When we make the RNA molecule, every 3 nucleotides is called a codon. And each codon will code for 1 amino acid. So this is going to give us our polypeptide or our protein sequence. Now, for the protein to function properly, it has to have the right amino acids in the right order, and it has to fold in its correct conformational shape so that it can do what it's supposed to do. Now, let's suppose we have a DNA mutation that changes the adenine-thymine pair into a cytosine-guanine pair. That will be transferred into the messenger RNA sequence, and then that will change that serine amino acid into a proline, or whatever different amino acid. So this is an example of a missense mutation, also called a substitution mutation. Substitution mutations mean that we change a nucleotide in the sequence, which can alter the amino acid sequence. But point mutations can also be insertions or deletions. So let's suppose we insert an extra nucleotide into this sequence. Now we are not just changing this one codon in the messenger RNA sequence. We have shifted all of the amino acids after that. So now every single amino acid after that mutation, after that insertion, will be wrong. It will be different from the actual correct protein sequence. We call this a frame shift. And a frame shift can occur with an insertion or a deletion. If a nucleotide was removed, everything would shift one nucleotide over, and essentially the same thing would be happening. The reading frame would be off, which would alter the amino acid sequence. So let's suppose we had a sentence. The cat ate the rat. This is like our codons. Every codon codes for a word that has some meaning. And then this sentence is logical and makes sense and has a function, kind of like a functional protein. What would happen to this sentence if we had a substitution and we changed one of the letters? We would change the meaning of the sentence. Now, this could still be a functional sentence. The bat, maybe, can a bat eat a rat? Maybe. So the sentence is now different. It's wrong. It is basically the wrong sentence. It would be like the wrong protein. What if we had an insertion? Now this is wrong from that whole extra inserted letter. It shifts all the other letters, and you can see the sentence is now totally messed up. The same thing with a deletion. If you lost a nucleotide or a letter, you lose the C, everything gets shuffled over because it has to be in 3-letter sections. Now this is completely messed up as well. So point mutations can either be substitutions, insertions, or deletions. And then the insertions and deletions can cause frameshift mutations, meaning the entire protein could be wrong. So sometimes when we change one letter, if we have one substitution, it might be that that protein could still potentially function. And we do accumulate many mutations over our entire lifespan in all of our somatic cells. So the next type, the next category of DNA mutations is recombination mutations. This is when we have large segments or sequences of DNA that are inserted or removed from a specific location in the genome. And now we have these sequences called transposable elements. They're also sometimes called transposons or jumping genes. And this means that these pieces of DNA can move around the genome. The genome is all of the DNA in one single cell. So if we have a piece of DNA that can move around, this transposable element, it can move around in 2 different ways. In one way, it can copy itself and then move and paste itself into another place, or it can cut itself and paste itself. So this is actually a normal process. All living organisms have transposable elements, and bits of DNA move around the genome all the time in a normal way. And this is beneficial because it contributes to genetic variation. Remember, all living things need diversity. We need to have biodiversity and genetic diversity. So transposable elements contribute to that. But what happens if it goes wrong? If a piece of DNA copies and pastes or cuts and pastes when it's not supposed to? So we'll look at the possible consequences of transposable elements. Here we have a sequence of DNA. This red region is a transposable element, or also called a transposon. So this piece of DNA will be several nucleotides long, maybe even a portion of a gene, or it could even be a whole gene. And over here we have a normal gene sequence, and there's a few things I want to point out. When we have a normal gene, there are coding regions and there are non-coding regions. These non-coding regions are called the introns, and the coding regions are called exons. So when we transcribe this gene to make a messenger RNA molecule, the introns will be removed. The yellow regions are showing regulatory regions. These are not transcribed. They're important for telling the polymerase protein where to bind to start transcribing a gene. And then at the end, it tells it when to stop transcribing that gene. So these are regulatory sequences. Now let's look at 3 different scenarios if this transposable element were to insert, number 1, into an exon region. What will happen? This exon region now has a giant, huge insertion. So that is going to affect the whole rest of the protein, and it will disrupt the functioning of the protein. You can't insert multiple nucleotides into the middle of an exon and have the protein still function. So this will form a non-functional protein. Over here, scenario number 2, this transposable element can insert into the regulatory region. So this means now the amount or the timing of expression of this gene is now going to be wrong. The protein sequence itself is not mutated, so the protein will be functional, but the regulation of the protein will be wrong. So the amount or the timing will be affected. Now, in this third scenario, the transposable element is inserting way downstream of the gene in a non-coding region that is not going to affect this protein or the regulation at all. So this will not affect this gene. So this is a silent mutation. There are a few different ways that we can have a silent mutation. We can alter the sequence of the DNA without actually causing disease or a noticeable impact on cell function or protein function. So the different ways that we can have a silent mutation would be if the mutation occurred in an intron, which would be removed. If it occurs in a portion of the DNA where there are no genes that are coded. If it changes a nucleotide sequence so that the codon is not altered. Let's suppose we have a point mutation and we make a substitution. Sometimes different codons will code for the same amino acid, so you can change the DNA sequence without actually altering the protein at all. If the mutation occurs in a gene in a cell type that does not normally express that gene, that will be a silent mutation. So remember that hemoglobin, for example, is a protein that is expressed in red blood cells. If you mutate your hemoglobin gene in your liver cells, that's not going to have any kind of a detrimental noticeable effect. If we have a look at this normal sequence, when we have a gene sequence, we code for a messenger RNA sequence, and then there are sometimes redundant codons. So UUG also codes for leucine. So sometimes you can have a mutation in the DNA that does not alter the protein sequence. So this is a silent mutation. And these kinds of changes also contribute to phenotypic variation. If you looked at, say, a sodium-potassium pump gene in humans and chimpanzees and mice, you would see that they all have functional sodium-potassium pump proteins, but their gene sequences are different. And that's because sometimes mutations can accumulate if they don't have a detrimental effect. So mutations are the ultimate source of genetic variation. That's where new alleles come from, where new phenotypes come from. So if we have a look at this example of bees, we can see that all kinds of DNA mutations have occurred that were not detrimental to the survival abilities of these bees. And so now we have genetic variation. Different phenotypes might be more or less beneficial in certain environmental situations. And then we have survival of the fittest, or evolution. So where do DNA mutations come from? Why do they occur? Sometimes mutations occur during DNA replication. Polymerase adds about 1,000 nucleotides per second as it is replicating DNA. If it puts the wrong nucleotide in, then that will be a mistake. Or if it adds an extra nucleotide, or if it skips a nucleotide. A lot of mutations occur during DNA replication. Transposable elements. They can move, pieces of DNA can either move to the wrong place, or they can interrupt a gene, or they can interrupt the regulation of a gene if it doesn't move in the correct way. Then there are substances that cause mutations. Mutagens will mutate DNA. Carcinogens mutate it so that it causes cancer. There are viruses that can mutate DNA. Viruses, when they infect a cell, they inject their DNA, or they go into the cell, they're endocytosed into the cell, and that DNA can sometimes be incorporated into our DNA. Probably most of the DNA in our genome that is non-functional now but is there and we don't really know why, probably came from viruses throughout evolution. And then we can have mutations that we've inherited. So some things can come from our parents, from the sperm or the eggs. About 10% or less of diseases that exist, including cancers, are from inherited mutations. Most mutations occur during an individual's life, and most mutations only affect the individual, and they're usually not passed on. So let's look at a couple of examples of viruses. So viruses that can insert DNA into our genome and potentially cause cancer or other diseases would be HPV, human papillomavirus. That's responsible for warts. That can cause cervical cancer. A couple of the strains can cause cervical cancer. HIV can cause Kaposi sarcoma, which is a type of cancer. Hepatitis B and C can cause liver cancer. And mono or Epstein-Barr virus can rarely cause a kind of lymphoma. So different kinds of viruses can cause diseases by causing recombination mutations or insertions that act a little bit like transposable elements. And here's a few examples of some inherited DNA mutations. Cystic fibrosis is a single substitution of a nucleotide in the gene that codes for chloride channels. Huntington's disease is caused by an insertion of multiple CAG repeats. Typically, more than 40 repeats will result in a disease that affects the brain. Sickle cell anemia is a mismatch or a substitution, these are the same thing, in the hemoglobin gene. Then the hemoglobin can't fold properly and it forms that sickle shape in the red blood cell, and then red blood cells are less likely to be able to carry as much oxygen. With cancer, there are usually 2 or more mutations, and they usually affect either the cell cycle or DNA repair enzymes. The 2 main categories of genes that affect the cell cycle are oncogenes, which will increase cell division, and tumor suppressor genes, which are normally supposed to decrease cell division if there's mutations. And if they're mutated, then mutated cells will continue to replicate. Phenylketonuria, or PKU, this is often tested for in babies. This is when a liver enzyme gene has a mutation, a single point mutation. Different point mutations in that gene can cause the same disease. And lastly, non-polyposis colorectal cancer is a type of repeat, a CA insertion repeat, that affects a DNA repair enzyme. So sometimes point mutations can cause disease, and sometimes recombination mutations can cause disease. Now, how does our body deal with mutations? What can we do about it? Do we just accumulate mutations for our whole life? Actually, we a little bit do, but we can get rid of a lot of mutations. For example, when we're replicating DNA, polymerase is the enzyme that is adding the complementary nucleotides as it's replicating. If it adds an extra or it puts the wrong nucleotide, let's suppose it puts 2 purines together, the polymerase itself can have proofreading ability. It can notice mistakes while it's replicating. And then what it can do is it can move backwards, and then it has exonuclease activity. It's a type of enzyme in that protein that can cut out a sequence of the DNA that it just added, and then it will go forward again and re-add. So when it is adding nucleotides during replication, it's moving in the 5' to 3' direction. Exonuclease activity happens in the reverse direction. So polymerase itself can help get rid of a lot of those mutations that occur during DNA replication. Next, we have DNA repair enzymes. We have about 130 different DNA repair enzymes that will look at the DNA and find mistakes. Sometimes it may not know which strand of DNA is correct, so they're not 100% going to remove all mutations. But DNA repair enzymes will notice when purine-purine combinations occur or when pyrimidine-pyrimidine combinations occur. And DNA repair enzymes can fix a lot of mistakes. Let's suppose polymerase doesn't notice the mistake and DNA repair enzymes can't fix the mistake. Now what happens is we have a mutated protein, potentially a mutated cell. then we have genes that can code for apoptosis signaling. Apoptosis is a programmed cell death, and this is when the cell basically kills itself because it has mutations it can't fix. There's a link below to another video that talks about that in more detail. Let's suppose proofreading didn't work, DNA repair didn't work, apoptosis didn't happen, and now you have a mutated cell. We actually have one more redundant measure, and that's our immune system. Immune cells can attack mutated cells, like cancer cells. And then if all of those mechanisms fail, then if you have a non-functional or a misformed protein, then you can end up with a disease.